Hello and welcome everyone in another episode of Researchers Celebrity. Today, we are delighted to have Professor ANISUR Rahman KHUDA BUKHSH with us as our distinguished speaker for this series. And a brief introduction. There are no words with which I can introduce Professor KHUDA BUKHSH to all of you. The people who are working in the field of homeopathy and the people who have followed his research already know, but for general audience, I will still make the introduction. Professor has done his bachelor's from KN College, Calcutta University, and then for master's and PhD, he attended University of Kalyani, Kolkata, and joined like after his PhD, he joined as a faculty. He has served in various roles and as was retired professor and emeritus of University Grant Commission, Government of India at University of Kalyani, where he still pursues and guide students, motivates them. With this, we'll say thank you very much, Professor, for taking time out for this conversation, and we look forward for this conversation, which will be the stone turning point for so many next generations of researchers. Thank you, sir. So with
So,
this, we always say that how you decided way back then that you want to become a researcher.
should I share now?
Sure, please. I'll say to the audience that for today's conversation, we'll have the presentation mode where Professor Anisur will take us through the journey and we all can. Yes, sir, you can share the slide now, please.
So, first of all, I thank you very much. And also, I am indebted to my friend, Dr. Sridhivash Adok, for giving me this opportunity to have an interaction with some great audience here. So, actually, I had some very early research training. I was a student of the same department at one point of time. And in my final year MSc, I had cytogenetics and molecular biology as my special paper, where I received some special training on chromosomal preparations and study of insect and mammalian chromosomes. Then my dissertation study of induction of various types of chromosome aberrations, micronucleus testing, sperm head anomaly, mitotic index, etc., by some mutagens also formed a part of my dissertation in MS. So actually, that was the beginning to say that I was going to shape myself in some kind of research aid. Now, after doing some brief research in the Department of Zoology with another professor, Professor A.K. Shah, I worked on some chromosome aberrations induced by some mutagens in grasshoppers. But soon enough, I got a CSIR fellowship, that is a project that I signed with my more famous professor, Professor G.K. Manna. He was an internationally famous personality and well-known researcher at that point of time. And he sent a project for me to work on fish chromosomes. Now, naturally, the question you would ask me, why of all things fish? What is the interest behind fish? Now, it is very simple. Fish is the primitive vertebrate in the first place, and fish was considered as a very difficult cytogenetical material. And in a country like India, hardly there were earlier studies on some 10 or 15 species of fish before we took up this challenging project. The main reason of that was the technique was not properly standardized, and also because the fish chromosomes are naturally large number and very small in size. So it is very, very difficult to get the divisional stages arrested in them. For arresting the chromosomes, we had to use colchicine, which is a very toxic chemical. This alkaloid tends to kill fish within 2 to 3 hours. But the technique that was in vogue at that time actually wanted me to make them alive for 4 to 5 hours. That was through practice I learned that 2 to 3 hours of treatment do not really produce any kind of good result. So we standardized the technique and then we started doing work on teleostean fishes. So there were a great many problems. You have to go to the fishermen on boat to collect fish in living condition. I had to take 2 bucketfuls of water to keep them alive inside after catching them from either river or from the ponds. So that was some hazardous things to do because I had to collect the fishes, and I had to find out and identify the fishes on which chromosomal studies have not been done earlier. So these are some of the difficulties I faced, and I started doing my research for 3 long years. And then my PhD dissertation was submitted. It was entitled Study of Chromosomes in Teleostean Fishes. I produced some karyomorphological studies of 24 species of fishes, which at that time was a great achievement because, as I told you, it is very difficult to get the chromosomal picture. In some fish, we got 100 chromosomes, as many as, and we had to draw them with camera lucida. We had to take photographs of the plates, and we have to determine the number. In some plates, 101, some plate 102 maybe, some plate 98, but I had to point out the nodal number, and I had to determine the Twain number, that also through some meiotic study of n number of chromosomes. So when I joined the faculty the department as a faculty right after getting my PhD, I thought that why not take up some challenging jobs like the fish chromosomes? So I started to try procuring some funding. You can imagine at the time when I started doing this study on fish, I had only one microscope to work with. And with the same microscope, I started thinking, what else can I do? And I sent 2 projects of mine for my own funding. Then after getting one from the UGC, I started studying fish. I extended the data by studying not only the karyomorphological studies, but biochemical studies on non-enzymatic like hemoglobin, transferrin, plasma proteins, and enzymatic proteins, isozymes, LDH, MDH, XDH. So I can tell you that these were not studied earlier than that period. This non-enzymatic proteins of fish not known. Enzymes were not at all studied because gel electrophoretic techniques had to be standardized for studying the LDH and the gene expression studies. We made the study on more than 55 species of fishes, and I continued and extended the data on fish karyology to more than 100. You can imagine how much of endeavor I had to give to study. At one point of time, people used to say, "Oh, Dr. KHUDA has done the chromosomes of this. Why are you trying to just repeat this? This is already done. Go and see." And at that time, there was no computer. So everyone had to study from the biological abstracts and learn by memory which are the fishes already done, which are not. So all the time I had to identify the fish first, whether somebody has worked on it or not, or that will be repetition. Now then I started thinking of people could not think of chromosome banding on fish chromosomes. It was not done earlier in India. We were the pioneers in making banding studies in fish. We studied nucleolar organization, this nucleolar organizer localization. This is the part where secondary constriction of the fish chromosomes are located. So we were the first to work on chromosome banding in fish, annual localization, more than 55, 60 species, then C-band, centromeric heterochromatin. This is all different technique that we standardized. And G-banding, that was the time when act, actually in 19, end of 60s, the banding studies started in the world elsewhere with one success or two successes here or there. So that was the time when we ventured to attempt the CGNOR banding and we successfully did and published papers. Then I also tried to study the hill stream chromosomes, hill stream fishes. So when I went to the hills, I found some aphids associated with some insect like ants. So I thought when I come to the hills, collect fish, why not I collect some aphids and try to. But aphids were considered more difficult because the embryos from which the aphid chromosomes could be studied, produced, prepared, were microscopical in size. We had to look through microscope to find out the smallest of larvae to prepare slides. That also we standardized. Okay. Now, this area was totally unexplored. There also we made a mark, our laboratory made a mark. We almost studied aphid chromosomes of about 100 species from India. We were the pioneers in studying aphid chromosomes. All these have been not only published, but in very standard books, those references have been put in. Now, in Indian mites also, we made a little forage, but that is not what's talking about. Now, after this, actually, I tried something very different. Because of my training, as I told you, that in early phase of our research, I tried to understand the chromosomal changes associated with some toxicological treatment. So this was a new area for us where we actually started making some toxicological studies, experimental toxicology, and we studied different types of radiations and sonications on cells, nuclear DNA damages. These were also new at that period of time. So what we did is genotoxic effects of whole body X-ray radiation on mice, effects of some heavy ions, heavy beam ion on Chinese hamster cell, on cell death, effects of sonication. Sonicates, this vibration, that also breaks chromosome. Sonication on mice, fish, grasshopper chromosomes, cytotoxic, genotoxic effects of some poison, toxic substances like arsenic trioxide, mercury chloride, etc. And also we started studying the effects of some toxic effluents, industrial area from fish. natural habitat. We also tried. Now, suddenly, I had made an accidental foray into homeopathy research. That I like to tell you that I was leading a postgraduate team of students to Simla when one girl student fell from stairs and got injured, teeth broken, and it was early in the morning. No doctor was available, nothing. Only we had Arnica montana, one homeopathic drug, with us, and immediately she had red, short eyes, and we administered at this Arnica montana, and it dramatically ameliorated the red eyes. It was okay. She was not feeling any more vertigo, bleeding stopped. And so in I started thinking, how could I prove something which most people don't believe in? Homeopathy has its own limitations. So what are they? There are certain scientific challenges. Clinical efficacy, it is clinically seen that people are getting benefit, but nobody believes. They say that this is placebo effect. The concern, what are the concerns? Concerns are use of ultra high dilutions where existence of drug materials doubtful, not only doubtful, but actually impossible. When you exceed 10 to the power minus 24, it exceeds Avogadro's limit of dilution, and you cannot get a single molecule of the original drug substance. You dilute it with 10 succussions or jerks like this, and then it is potentized or dynamized. And you take 1 ml of from 100 ml and put again 99 ml to make it 100 ml and again give 10 jugs, it is potency 2. So each time you are diluting it to the power 10. Okay. Now, this mechanism that was not at all known. Nobody could explain how something which does not carry any drug molecule at all physically, how could it work? They don't believe in the homeopathic drug at all. And similar principle, great Samuel Hahnemann, he was an allopathic physician, but he discovered more than 200 years back the homeopathy. It tells that like cures like. Means if you consume raw arsenic trioxide, it's a poison. You will die after vomiting, after diarrhea, and after toxic symptoms will appear within a few minutes and you die. But if you see the symptoms in a normal, healthy person, this symptom of diarrhea, symptom of vomiting, symptom of weakness, no feeling appetite, burning sensations, all symptoms of arsenic trioxide. And if you administer the diluted potentized form of arsenicum album, then it acts as a remedy for the arsenic toxicity. So, understanding this similar principle, how could the cells react to a substance which is toxic in nature, one given when given in greater concentration dose, the same thing when potentized and diluted, absolutely there is no molecule present, only the molecular imprint, they act as a medicine. So this similar principle, and also it can act on mind body. It also takes into account your mind, your nature of mind. Are you a miserly person? Are you a very, rugged person? Or so the mind body interaction also it speaks about. Now, after 3 nights, sleepless nights, I started to think, what if I do some experiment on chromosomes? Now, which mice are so little. If I hit it, it will die. But I have to get some kind of shock and injury. And how can I do that? Then, as I told you, we had some experience of this radiation damage earlier. Suddenly it came to my mind that I can do it very well. I can expose the mice whole body to whole body X-ray radiation at a sublethal dose. Very small amount of dose which will not kill, but it will affect the chromosome. That we have seen. It is it is making change both quantitative and qualitative. I can measure, and it is dose dependent. So after that, I immediately thought that we are going for this experiment. So mice genome, they are very close to human genome, and it is a nice model for studying this kind of thing. So what we did in brief is we had to be very precise and make an experimental design which will be acceptable to all. And we had to make some studies which are very much alive at that period. It is anti-radiation action. If I can show that with controlled trials, that the homeopathic drug in minute doses can make certain favorable changes in the chromosomes, then people will have to believe. And the chromosome breakage and DNA repair, there is a very elaborate mechanism. At that time, it was known and established. So what we did is we have kept several controls. One is diluted alcohol, only water. One is diluted unsuccused alcohol. One is placebo, succused alcohol, the same way diluted. And Arnica montana, one, I mean, below Avogadro's limit, 630, 200 C. Then Hypericum, Ruta, etc. These are all known to be against shock and injury. They give treatment to someone having some kind of shock and injury. So we elaborated the study with pre-treatment of drug, only post-treatment of drug, pre and post-treatment, before radiation, then after radiation, drug, before radiation, only drug, before, after, only after radiation, like this. So we tried Arnica montana, Hypericum, 3200C, Ruta graveolens, etc. These are all known to protect the chromosomes. And now we have seen that the chromosome aberrations decrease. Yeah, I have here. Thank you. I have here. So chromosome aberrations decreased, repairs done. Placebo or succused alcohol or all other controls, they could not bring these changes. So we thought, this is very great thing. When we studied the micronuclei, these are broken chromosomes being extruded. So we have found that this micronucleus number was reduced. Sperm head anomaly decreased, protected the sperm. And also mitotic index, that is increased. More number of cells being produced. So the damage is being replenished. So this was something which simply agitated us and stimulated us to perform much more with great parameters, because we knew that chromosome repairs could not have been done without the genes in mammal XPA, XPV, XPC, XPD. These are some of the genes. Someone will recognize where there is damage in DNA. Helicase will open up DNA binding. Then nucleus 5' nucleus, it will cut that damaged area, and dimer So the entire mechanism must have been stimulated and triggered. Now I thought, what about some toxic chemicals? Hahnemann told like cures like. So if arsenicum album could protect again, if Arnica montana, which is used against shock and injury, protected, then arsenic trioxide, if I use, there must be certain genotoxic changes. Yes, there was. And arsenicum album prepared from tiny doses that protected. Mercury chloride, it was given Mersol 30C, that protected. Cadmium, that protected. So we confirmed that this principle, if we follow, something we treat with the same poison diluted, then there may be some kind of protection. So when I published it in the international journal BMC Complementary and Alternative Medicine, after 6 reviewers, different reviewers checked the manuscript, and different people asked me to defend their queries. I did it for thrice, 3 times. They were not satisfied. Then the chief editor said, look, there is a point where we should stop. And they all agreed that this kind of study is very rarely done and it is a mammalian model. So let us publish it. And when they decided to publish it, they added one international press release that look, one Indian group tells that arsenicum album prepared from very low dose and suckers violently, they can prevent some liver toxicity enzymes, modulate enzymes, they can make certain favorable changes. So it is something very curious. What is your opinions about it? Immediately, new scientists made an interview with me and they were excited to know that we have done it through double blind check. We have used statistical analysis in it. Then BBC News, they focused on this remedy cures arsenic poisoning. Science DevNet, and in many leading, and this paper was considered as an age paper of 2003. And then, when I was starving for funds, when I had no fund to do biochemical works or major works, I was only depending on our government funds, which I wanted to do some research on, say, effluents, say, aphids, say, fish. I got the chemicals from, and we there were a flow of funds coming from various countries. They wanted to make collaboration with me. But they made strictly one condition that this study, human trial, have to be, uh, you know, uh, double blind. And I could not agree to that. An American group wanted to know equal number. Because when I visited the groundwater arsenic village, I was simply stunned to see the miseries of the people and the condition of the people. They were dying. Lakhs of people were dying. And because of this work, my name was placed in Wikipedia. They mentioned about the work. And also, I was considered one of the zoologists, distinguished zoologists of the 20th century. Now, in 2 countries, adjoining countries, Bangladesh and West Bengal, there is a menacing problem of groundwater arsenic. Lakhs of people, crores of people are affected. Only in Bangladesh, more than 2.5 lakh at that time died of arsenic poisoning. And in West Bengal, 9 districts were already affected. And there were no practically modern medical treatment for arsenicosis. And arsenicosis patients only don't take water, poisoned water, but who is going to give them ready arsenic-free water? That's a great problem. Now you can see that these were the pictures. When I went there, all people are having various kinds of rashes. Then I have, because the time is short, I could show you some very, very bad pictures of tumors, bad pictures of some, boils. And so then we started this, decided I must do in 2 ways. In the first way, I have to satisfy science. So in one lesser affected group, I made a urinary arsenic acid survey. What is the arsenic content? And whether after administration of arsenic amalgam, whether the arsenic is going out or not. So I had to learn about the assay of arsenic. Now you can see that after giving the arsenic people, they are frustrated, they cannot work, they cannot sleep, they have burning sensations, and this. You can see that there is change, positive change. Now, arsenic content of urine, blood of over 150 subjects of the arsenic contaminated areas were surveyed, and many positive alterations, various toxicity biomarkers. These are all liver biomarkers. Some are triglycerides, some are so positive effects of autoimmune system. You know, it's dangerous. ANA positive people we have got enough. So arsenic album was working on ANA, anti-nuclear antibody also. All standard scientifically accepted protocols were followed. So you could see that they were gradually actually getting reduced within 45 days. Now, other modulations, glutathione, blood parameters, then matrix metalloproteinase, immunoblots of BCL2, positive modulation, ANA titer, etc. So, what could be the clinical benefits in those remote villages of taking arsenic amalgam? External arsenicosis symptoms reduced, appetite increased, gastric problems better, energy spirits increased, desire to live less frustrated. The people are frustrated, they wanted to die. Those who could not sleep could now sleep. Those who had burning sensation had less burning, muscle joint pain, and improved strength to work. They were sitting idle and waiting for death. And at one village, we did not find a single person aged more than 45. All people died after 45 years of age. So we were thrilled and started to do another human trial with the grant that I got from Boiro Laboratory. At the time when I did not have any grant from India, 4 years, I did not have. That was a period of frustration. I could not do much work. But I got a grant from England. Thompson, Dr. Thompson was one, and he was kind enough to grant me only 1,800 pounds to outsource some of the work on this arsenic. He was very much interested with arsenic. So I used to send them materials, but they could not because of this. They could not do much there. What I did is we carried on the study to thalassemia patients. You know, thalassemia is a gene mutation that is responsible for failure to produce beta chain of hemoglobin. This is a almost deadly disease at one point. If it is diagnosed early, then you transfuse through blood transfusion and hydroxyurea allopathy drug is there. But after certain period of time, they are prone to die. So we thought that what if we could delay their death, we could delay transfusion, and we could delay this big belly that is spleen enlarged. So we tried pulsatilla, sionathus, ferromet along with hydroxyurea. And the result was fantastic. Within 45 days, again, this ferritin level increases. I mean, in the blood, only some chelating agents can be used, but that does not make any difference. Blood, iron deposition is there, and people die. Now, spleen enlargement is there. So we tested various glucose and various parameters, and what gains? Distinct fall of ferritin level in blood serum, only with treatment of homeopathy plus hydroxyurea. Decreased size of spleen, 11 patients showed absence of palpable spleen after homeopathic treatment. As many as 11 out of 13 patients, 38 patients, they did not have spleen at all, palpable spleen. They were cured of that. Gaps between transfusion time increased, general improvement in health and spirits, no fever during transfusion in 2 patients. Now the big question, can it work on cancer? Another challenging problem of the world. Now, we started making a probe as to how we could induce cancer, liver cancer, lung cancer, skin cancer, and after study, we found that yes, PDAB and phenobarbital, these are the carcinogens which induces liver cancer if chronically fed to mice. Then lung cancer also could be induced with one carcinogen, and skin cancer with DMBA and another aryl hydrocarbon receptor, one that is proton oil, I think, rubbing. So these three models we utilize. These are very good models because we could, when we dissected open, we could visibly see the cancerous growth in form of nodules, tumors inside liver. And if homeopathy drug could cure, there was either less cancer load on liver tumors or no load, or also the skin cancer was a better model in the sense that at the time when there was treatment going on, we could not see what is the condition of liver in the interim period. But in case of skin cancer, it could be visible that papilloma was growing. And when I was giving sickle core to a group of drug-treated ones, there was less number of papilloma, regression of papilloma. So this we started doing. So with all these, we conducted controlled research utilizing an array of widely accepted modern scientific protocols, tools like cytogenetical, DNA fragmentation assay, comet assay, biochemical, histological, histochemical, enzymatic, hormonal, gel electrophoretic, electron microscopic, spectrophotometric, physiochemical, fluorescence, facts, immunoblot, nanotechnological, producing convincing and repeatable results. People don't believe us, but we had to make them believe because all these papers were published in, say, Colloid and Surface, a big journal, Science of the Total Environment. This is having, at that point of time, more than six, seven perhaps. So results of these studies demonstrated efficacy of ultra highly diluted potentized homeopathic drugs, diverse models. Then we started foraging in other areas. What if we treat it to bacteriophage, that is virus? very dangerous. This is virus, bacteria, yeast model, mice model, rat model, humans, and in vitro cancer cells. When the cells are only there, detached condition. Can homeopathy work on that? And we tried to focus on the mechanism of action. Now, study parameters as follows. So we have we have published many papers. So people interested in this can see that, and we have been able to show that we got positive results. Now, I only mentioned two, three very interesting experiments. One is E. coli intoxicated with sodium arsenide and arsenicum album was given. In fact, arsenic trioxide or sodium arsenide, the pathway or how smaller animals or prokaryotic animals like bacteria can protect themselves from natural exposure to these was being studied. There are arsenic pumps. Arsenic pumps, if arsenic by chance enters, it will prevent arsenic to entry. And even if it enters, then the pump, arsenic A gene, arsenic B gene will be triggered. And these genes triggering can be studied and quantified by studying their expression pattern. So what we did is we know how arsenic tries to, at the expense of ATP conversion to ADP, they are pumped by the arsenic AB gene. We also know that ATP is very much required. So glucose is very much required. And we also thought that glucose is required, but we will not give glucose in the medium. We give them in the potentized form of glucose. We succus, make it potency 1, potency 2, potency 3, and when it crosses 12, then 30 potency, 10 to the power minus 60. So there will be no trace of glucose. And then arsenicum album were given to arsenic exposed bacteria, E. coli. And the thing happened is the ARS-AB gene was triggered. They were triggered. And we measured the activity of the genes and they triggered. We also studied the amount of arsenic inside E. coli and amount of arsenic given initially in the medium so that we could understand whether they have prevented arsenic to get entry or they are helping to tolerate arsenic whichever has entered. Now, in the E. coli, which has been exposed to sodium arsenide, we also gave glucose 30C. Oh my goodness, we found the glucose activating gene was triggered. And there was more of glucose, giving supply from inside the cell to the formation of ATP. We measured, quantified the ATP, and we found that one was doing in one manner, triggering one particular gene, and glucose 30 was triggering the glucose activating gene permeates. So that was a marvelous, exciting finding that even E. coli could distinguish and trigger specific genes which are relevant to the environment they have been exposed to. So, glucose 30 promoted more glucose entry by activating relevant glucose uptake, and we made quantitative studies of ATP, gene expression studies, etc. Now, another is also very striking, which I could not cut down from my talk. You know, there is a fudge, bacteriophage. This is host specific to E. coli. So we had already made one laboratory for E. coli study and this study. This fudge attacks the bacteria and it will leave its shell, protein shell outside, inject the DNA inside, and it will start constitutively, expressing the E gene, which is a lysis gene. So after getting incorporated into the bacteria genome, they start producing more and more mRNA. And that produces the protein required for replication of the fudge. And this fudge, when they are given a tray, they will make these plugs where they have free growth. They have eaten up this growth medium and produce these plugs. So number of plugs you can see that tells you about whether the mixture, if the bacteria is giving some homeopathy drug, they can protect the formation of the plug. And you can see that this is control where nothing has been given, only food. This is the placebo, suckers alcohol given, but the same number of plugs. Arnica has little less, but Belladonna has great. Rustox has great anti and aconite. This has also some antiviral activity. What happens is when we search for the specific gene that has been stimulated in the bacteria after administration of these drugs is the gene that is restriction endonuclease that cuts the DNA of the virus. And that is the reason they are not functional anymore. They cannot eat this anymore. So, significance of the result is potentized homeopathic drugs show antiviral effects in E. coli. Is a virus, basically. E. coli can distinguish ultra highly diluted homeopathic drugs and respond differentially in respect of their gene expression. E gene in 5174 turn on being constitutively expressed when inside E. coli, and artificial studies showed altered level of mRNA expression. So, then again, we have done a lot of studies on, I now skip, there are molecular studies made on diabetes, cancer, then apoptosis. These are it's the same kind of study extended. Now, overview of the works in different models. We have done these works. And now a new chapter begins. How the homeopathy drug, can they be nano-encapsulated or nano-precipitated by homeopathic drug? So at the one stage, we encapsulated the semi-dried or dried up mother tinctures into PLGA nanocapsules. And in other study, we tried to see whether the mother tinctures, they were capable of precipitating nanoparticles from silver nitrate. These we have published in very big journals. And we have tested this in so many, drugs. And these are the physicochemical characterization, chemical characterization we have done. And all these we have used for ascertaining that we are correct. The DNA CD studies, we have shown that there is conformational change in the DNA after administration of homeopathic drug. We have molecular docking studies. So the modern techniques, then nanoencapsulation. What we wanted to say is that when we suckers and prepared the dilutions. They reside, their molecular imprints reside in nanoparticle form, particulate form, and nanoparticles were later discovered by Bilare in IIT Mumbai. These have been published. Okay. Now, in brief, the significance, drug with equal or more stronger effects. Let's, when we nano encapsulated, very small, but they are more powerful. So it goes with the principle of homeopathy. Small size nano encapsulated drugs have zeta potential negative, so they can quickly enter. PLGA non-toxic, easily degradable. It can be used as a carrier. The drugs were found to be effective against cancer, diabetes, and more similar drugs should be done. Now, with all these backgrounds, now we came to the conclusion that ultra low doses of homeopathic drug must act through regulation of gene expression. It works at the molecular level. They act by triggering master genes as though you arrange some bricks one after the other, one after the other. You push the first one, the whole cascade of chain reaction will follow. And modifications they bring may be affected through drug DNA interaction, resulting in epigenetic modification, bring about phenotypic changes with altered expression of gene orientation without actually altering the original DNA sequence. That is a remarkable findings. And in proof of that, we have published this DNA methylation, demethylation, histone acetylation, deacetylation of gene expression. This has been published. This is the first time we have done that. This is a sure sign that the DNA is involved after administration of the homeopathic drug. Then DNA microarray. For one experiment, we had to spend more than 5 lakhs of rupees. And we have found that these two drugs could modulate the expressions of 122 genes, upregulated and downregulated some, and among them 60 very pronounced and prominent cancer genes were involved. And we are working on the effect of anti-cancer gene expression. And so we propose this gene regulatory hypothesis, which we have given many examples and proofs from our own experiments. And these are all definite proofs of the genetic action, gene expression action, which is caused by, and that is what the whole of world is pondering over. And recently, one very famous scientist in PLOS paper, he wrote that perhaps the hypothesis given so far by all authors of these most tenable and practical working hypothesis is that of KHUDA, which is very likely the main way how the homeopathic potentized drugs could act. So I think the take-away points, these are for the scholars of nowadays. To me, every research is important in the advancement of science, whether fundamental or of applied value. So enjoy doing it. If you don't enjoy, don't do it. One can utilize whatever laboratory facility, financial resources are available while trying for procuring funds. Funds is a very difficult proposition. The competition is great. And nowadays, the funds are dwindled and more of hands are towards it. Well-designed experimental research that has implications or practical applications get extra prominence for its greater impact on life and society. I admit. But innovations and explorations into different domains can bring more interest, diversity, and excitement in research. But carrying out in depth studies in a particular area has its own benefits of making further advancement of scientific knowledge in that particular area. Both are meaningful. One thing is common to all: there is no substitute to hard work, diligence in getting success in a research career. So these are some of the things. After so many years of research, every researcher faces failure, ups and downs. Everyone had some kind of difficulty in lab facility, in instrumentation, so on and so forth. But if you have the zeal, if you are not afraid of giving hard labor, and if you are fond of doing both laboratory and outdoor research, then you are in yourself satisfied at the end of the day. So, with this, I thank you all very profusely for giving me this opportunity. And I hope I have not crossed much time, but I these are my students, these are the students, and I have collaborations that I miss. I many universities wanted and worked. I just একবার দেখিয়ে দাও. These are Calcutta, Jadavpur, Bardhaman, JNU, Kanpur, IIT, then Kalyani, Mohit Bhattacharya, SPJ, pharmaceutical chemistry. Then there are so many Harvard and University of California, Riverside. So I have collaboration with so many people. They actually wanted to collaborate. Some people made some chemical sensors. They wanted me to see its action in living cell. So whether they are active in living cell or not, how far they are sensitive to the particular compound or say phosphorus sensors they are prepared. I've shown how it is showing phosphorus inside cell when we add to it. Or so this kind of collaboration also I have done a lot. So this is the material outcome is nearly 400 research articles, including reviews, book chapters, research papers I have published, we have published. I have mentored research of 51 PhD scholars so far. Google citation more than 10,050 or like that. Research gets read more than 2.25 lakhs, but citation is not great. And H index, my present current is 56, I'm nearing 57 in a few days maybe. And this 4288 research interest score is among the top 0.5% of world scientists. So from a very limited facility, small university, if I could do this kind of things, more talented students, scholars, nowadays, they can do much more. And I have been given, in recognition of my Research Lifetime Achievement Award from Government of India. This is a great glory for me. Lifetime Achievement Award from European Committee for Homeopathy, consisting of 26 countries. They have chosen me, an Indian, to give this first award. And I was finding a gala arrangement in Vienna town hall where I was felicitated and given this. Other than I have in Stanford University race, I have been in the top 2% for the last 5 consecutive years. World scientists scan this area. Career impact from 1960 to 2022. My rank, global rank, is considered to be 26 globally and rank 1 in India. This is given by Stanford University. And for the last consecutive 5 years, they are mentioning my name as one among the top 2% of the world scientists. So I am grateful and thankful to God Almighty. And I am also thankful to the financial agencies who did it possible to carry on research, and my students as well, who actually sweated and sweated for doing this kind of research in multiple disciplines, covering various modern and backdated techniques. All they have done by themselves in our own laboratory, most, and in some cases we had to offshoots, of course, like electron microscope. We did not. So thank you, and I am very much grateful to all of you. Thank you very much.
Thank you very much, sir. And to conclude, I'll say to all the researchers who think that now we are talking about interdisciplinary research, Dr. Khuda Baksh has done this like 5 decades ago. And for all who think that you are limited to one organism or one model system, Dr. Khuda Baksh has done his work from fish to aphids to mice to toxicology and then thalassemia, cancers, and diabetes, whatnot. He has developed tools which people are using now and getting benefited. All the recognitions which he got, lifetime achievement awards, are nothing in front of the lives he has saved and the impact he has made. With this, we will say thank you so very much, Professor Khudabaksh, for sharing this journey and motivating the next generation of researchers. For all the audience, I'll say stay tuned for another episode of Researcher Celebrity. And please feel free to write to Professor Khudabaksh or to Empowering Science Foundation if you want to be in touch with our Researcher Celebrity or want to be mentored by them. Thank you so much, sir. And for researchers, stay tuned for another episode of Researchers Celebrities.